Hello friends, good morning. Welcome to Sam Biology class. Friends, today we shall discuss about bacterial conjugation. In my last video tutorial, we discussed about transformation. Transformation doesn't occur in E. coli. Then the question is, if there is any kind of gene transfer in E. coli, the answer is yes. In 1946, Joshua Lederberg and Edward Tatum discovered that E. coli transfer genes by conjugation. Bacterial conjugation, also known as mating, is a process of horizontal gene transfer by which genes are transferred from a donor bacterium to a recipient through a specialized secretion system. For gene transfer to occur by conjugation, a close physical contact between the donor and the recipient bacterial cell is essential. Donor bacterium have cell surface appendages called as pili, singular pilus. On the basis of function, pili can be divided into two groups. Attachment pili that help in the attachment of bacteria to different surface, and the other is conjugative pili or sex pili that mediate conjugation. Its diameter is from 6 to 7 nanometer. Perhaps the most well studied example of sex pili is the F. pili of E. coli. The synthesis of F. pili is controlled by genes that are present in a circular double-stranded DNA molecule called as F factor or sex factor or fertility factor. F factor is a plasmid. It contains all the necessary genes that are essential for replication and transfer. The F factor can exist in two different states. One is autonomous state and the other is integrated state. In autonomous state, the F factor replicates independently of the bacterial chromosome. This is a bacterial cell. This is the bacterial chromosome and this is the F factor. If the F factor exists independently of the bacterial chromosome, then that type of bacterial cell is called as F plus cell. Sometimes the F factor can also be integrated covalently into the bacterial chromosome by site-specific recombination events. This integration is mediated by short sequences that are present in multiple copies in both the bacterial chromosome and the F factor. So, the F factor can be integrated at many different sites of the bacterial chromosome. So, the bacterial cell which contain an F factor either in autonomous state or in integrated state can act as donor, so it can transfer genes to a recipient bacterial cell. Friends, as already mentioned, the HFR strain is produced by the integration of the F factor into the bacterial chromosome by site-specific recombination events mediated by IS elements that are present in both the F factor and the bacterial chromosome. But the question is whether the integration of the F factor into the bacterial chromosome is irreversible. The answer is no. Presence of rare F plus cell in the HFR culture prove this. Sometimes the integrated F factor is excised from the HFR chromosome. When the excision is abnormal or anomalous, that means the integrated F factor is excised at wrong place, then The excised F factor may carry some of the bacterial genes. Such a F factor that carries some of the bacterial genes are termed as F prime factor, and the bacterial cell that contains such type of F factor are called as F prime cell. So there are three different types of donor cells. Number one, F plus cell, number two, HFR cell, and number three, F prime cell. All three of them can transfer genes to a recipient. So friends, now we shall discuss about three types of mating or conjugation. They are F plus into F minus mating, HFR into F minus mating. Third one is F prime into F minus mating, which is known as sexduction or abduction. Conjugation is responsible for horizontal gene transfer among bacteria. It is the primary means by which antibiotic resistance genes spread among bacterial population. During conjugation, genetic materials, notably plasmid DNAs, are transferred from donor bacterial cell to the recipient. Conjugation is mediated by a type 4 secretion system that operate in both gram-positive and gram-negative bacteria. A conjugative type 4 secretion apparatus in gram-negative bacteria is composed of 12 components that are termed as VIR-B1, VIR-B2, B3 and so on up to VIR-B11 and VIR-D4. All these components form a multi-megadalton assembly. VIR-D4 act as the coupling factor. In addition to this, the type 4 secretion system also contains a F pilus that is composed of VIR-B2 protein, also known as pillin, 3 ATPases such as VIR-B1, VIR-B11 and VIR-D4 power the system. Now we shall discuss about F plus into F minus conjugation or mating. During F plus into F minus mating, a copy of the F factor is transferred from donor cell to the recipient through a type 4 secretion system. F plus cell which has a copy of the F factor is the donor, whereas F minus cell which do not have F factor is the recipient. First of all, the F pilus, which is present on the surface of donor cell, establish and contact with the recipient. Once contact is made, the F pilus retract, thereby bringing the donor and the recipient cell into a close physical contact. Replication of the F factor occur by rolling circle mechanism. Replication is initiated by protein complex known as relaxosome, which nicks only one strand of the double-stranded F factor at a specific site called as ORI-T or origin of transfer. After cleavage, other components of the relaxosome are released inside the donor cell. The other component, which is the enzyme relaxase, remain covalently attached to the 5' end of the nucleostrand. Then the relaxase DNA complex is recruited to the type 4 secretion system by the coupling factor. During the transfer of relaxase and the nucleostrand, the strand is replicated by rolling circle mechanism. In this figure, you can see that the nucleostrand has been transferred from donor to the recipient cell. During this transfer through the tie force secretion system, this entering strand is replicated, so the recipient cell receive a copy of the effector. At the same time, the remaining strand in the donor cell is also copied. So at the end of F plus into F minus mating, both the donor cell and the recipient cell have a copy of the effector. It is because of the transfer of a copy of effector from donor cell to the recipient also become F plus. The second type of donor cell is HFR cell, which is produced by the integration of the effector into the bacterial chromosome by site-specific recombination events. The mechanism of HFR into F minus conjugation is similar to that of F plus into F minus conjugation. First of all, the donor and the recipient bacterial cells are connected by a tie force secretion system. After that, transfer and replication is initiated. As the transfer is initiated at the origin of transfer, which is located within the integrated effector, a part of the effector is transferred prior to the transfer of bacterial genes. The remaining part of the effector is transferred after that. As the transfer of the complete HFR chromosome takes about 100 minutes in most of the cases, the transfer is incomplete. That means the recipient cell receives a part of the effector and a part of the donor chromosome. In this case, recombination occurs between the donor DNA fragments and the recipient chromosome. As the transfer is incomplete, only a part of the effector is transferred to the recipient. As a result, the recipient cell remains F minus. Only in rare cases when the entire HFR chromosome is transferred from donor to the recipient bacterial cell, then only the recipient becomes F plus. Otherwise, it remains F minus. So this is about HFR into F minus conjugation. The third type of donor cell is F prime cell, which is produced when the integrated effector is excised inaccurately from the HFR chromosome. So when there is error in the excision of the integrated effector, it may carry some of the chromosomal genes may or may not be in exchange of its own. Such a effector that carries some of the chromosomal genes are called as F prime factor, which was first identified by Edward Edelberg and Sarah Burns in 1959. The bacterial cells which contain the F prime factor is termed as F prime cell, which are rare in the HFR culture. The mechanism of F prime conjugation, which is also known as abduction or sexduction, is similar to that of F plus into F minus conjugation. First of all, the donor and the recipient cells are connected by a tie force secretion system. After that, transfer and replication is initiated. Transfer is initiated at a specific site called as origin of transfer, at which only one strand of the double-stranded F prime factor is cleaved. After that, the cleaved strand is transferred from the donor to the recipient bacterial cell through the tie force secretion system. During transfer, this displaced strand is replicated. As a result, the recipient cell receives a copy of the F prime factor. At the same time, The other strand of the F prime factor is also replicated to generate a complete copy of the F factor. So friends, this is all about conjugation in bacteria. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thanks for watching.